我们今天呢是邀请了这个来自加拿大的这个 Professor Northoff 教授呢，来给 talk. Before your talk, I will give a brief introduction. Professor Northoff is a philosopher, neuroscientist, and a psychiatrist. Originally from Germany, he was recruited to head up the mind, brain, and neuropathic research unit that is in Northoff. Professor studies the relationship between brain and mind in its various phases, focusing on the neural and biochemical mechanisms related to higher order mental functions like consciousness and the self in both healthy people and also in those with such mental illnesses as depression and schizophrenia. And Professor Northoff is one of the leading figures in linking philosophy and neuroscience. And he's also the author of 260 journal articles and 15 books. And so much more. I just stop here. Let's welcome Professor Northoff.
Thank you very much for the kind introduction and also the invitation. Happy to be here after 1 year. I think last year I was here at the Neighboring Institute and show you some of our approach and I specifically tailored also my talk to psychology. So the main idea, what I want to do is really how can we make the connection from brain to cognition or brain and mind. And I show you a novel approach which I hope provides a tighter linkage between the neural activity and your cognitive or mental. And that's what we call spatiotemporal neuroscience. So I start. Okay. So I start with an analogy with iceberg. So what can we learn from icebergs? As you all know, icebergs surface here. Okay, I do. Yeah, but you can barely see it. So icebergs have a surface and they have a deeper layer. So, and what we usually do, we investigate cognition up here and look for neural correlates of attention and memory. That's the upper part of the iceberg. That's the tip of the iceberg, what you see above the water. However, as you all know, icebergs have a deeper layer. And I say, the main argument, I go beneath the water, beneath what is usually visible, to the sort of invisible. And what do we find there? There we find the spatial features of the brain, topography, and the temporal features of the brain, stand up. So, and the main content of my talk is to show how these spatial temporal features, the dynamics and the topography, modulate cognitive function. And I will show this for 4 different features. For attention, that's what I call spatiotemporal neuroscience, and I will show this for attention, for memory, for thoughts, for consciousness, and if there's time, and I will also bring one application for psychiatric disorders. And at the end, I will make a conclusion about the spatial temporal model of the brain, mind, and cognition. And you already see the way I do it. I point out different kinds of dynamic features. Dynamic and topography are not homogeneous concepts, but they have a variety of different dimensions to them. For instance, one dimension of dynamics describes the change from time point to time point. The relative changes over time is variability. So here you see I need attention to variability. Then one line of research, what we're doing is linking thoughts to particularly the face dynamics, the ongoing dynamics of your face fluctuations. Then, of course, memory, no surprise, we link it to temporal features, to time scale. When you look into the literature of memory, that has rarely been done, timescales of the neuronal level. Then I show you some examples from our consciousness research about topography, and then I will make a conclusion with a couple of slides what this spatiotemporal model of brain cognition implies, and some more. So let me start with the first one, attention and variability. So what is variability? Variability is change. So you often observe this in your data series. You have reaction times, and you see a trial-to-trial variability, and you don't probably like this variability. You want to cancel it out, you want to average out, and you calculate the mean, and you might even exclude some of the reaction time data which have an SD larger than 3. Yeah, because it's noise for you. So you want clean data. However, what is noise for you may be the target feature for me. Because variability obviously has some technological artificial sources. In that case, it needs to be cancelled out. However, variability is an intrinsic feature of our brain. And it's manifest, as you all know, in your trial-to-trial variability of your reaction time, which is not often calculated. So now what we did here, and also you also know all our attention. When you're listening to me, you do not always pay the same level of attention, but it fluctuates. And you probably have not much influence on it. Yeah. So what we did here, we did a typical choice reaction time. We did a CPT task, continuous performance task in a way, and we did this in the auditory modality as well as in the visual modality to see that the same dynamic features hold in the cross-modal. So, and what was important, so here of course you get reaction times, and what is important that here is the time series of your behavior, that we bandpass filter the time series in exactly the same way we bandpass filter the fMRI scan. So we get the same time range of data in both behavior and neuronal. That's very important if you want to make a connection with that. And then we can link the amplitude of the reaction time fluctuation to the amplitude of the fMRI fluctuation. That's what I will show you now. So that's in slightly different ways that we look really into the time series. What you usually do, you take the time series and then you average your reaction time over the time series. But what we are doing, we're looking at the time series itself and looking how it varies over time. So this is somehow conceptualized as trial-to-trial variability, but it's it's your intra-subject SD, if you want to say. You can also calculate the coefficient of variation, divide the intra-subject variability by the mean. So, and then what we see here, these are now real data, and you'll see, again, you see quite nicely, here's the original time series, then we convert that time series into a power spectrum. So we have a power spectrum of your behavioral data. It's not a power spectrum of EEG or MRI, it's a power spectrum of your reaction time because we do a fast Fourier transformation. Yeah, and then we bandpass these data between 0.01 and 0.1 Hertz. Why? Because that's exactly the same bandpass as we use for MRI or CT altitude. So, and we do the same for the auditory task, and you see the fluctuations here, and you see also the slow frequency fluctuation, and then we also correlated the variability in the auditory task with the variability in the visual task. No surprise, we had a correlation. So that's nothing special. So now, in the next step, we go to the brain. So now the question is, are the fluctuations in the reaction time during that attention task related to the fluctuations in your neuronal activity? Yeah? So, now I need to push the right button. So then we analyze the task. As I said, we did the task also in the MRI. And here you see the activation pattern. So first we calculate the typical beta amplitude-based amplitude, and you can see the typical regions, no surprise. You see the attention networks here, dorsolateral frontoparietal attention networks. You see the typical regions, and we can make a beta value, and you see in both visual and auditory tasks. You see quite an activation, and you can see also quite a good transmodal overlap, particularly in the attention networks. So this is nothing new. You probably know this much better than I do. So then we also looked, okay, how about the fluctuations? So we calculated the neuronal variability and calculated for each task. And interestingly, we found in both visual and auditory tasks, we found a strong variability peaks here in the cortical midline structures, default mode network. That goes along with earlier work by Cucci et al., Cerebral Cortex 2016, I think it was, and some other papers. And we also looked for the transmodal overlap of the neuronal variability, and you could really nicely see it's the typical cortical midline region of the DNA. So one noteworthy thing, which I don't show here, you could really see that the neuronal variability involved different regions than the amplitude-based. Yeah? So this is a neuronal signal averaged across time. This is a neural signal showing the fluctuation across time. This is neural variability. This is the average across time amplitude-based measure. So now, of course, the next thing is what you really want now then is, are these fluctuations related to the task? So that's the first thing you want to see. Or are they just intrinsic fluctuation? If they're purely intrinsic fluctuation, you would not expect a difference between resting state and task state. But here we could really see that the fluctuations are really related to the task, because you see higher fluctuations in both auditory visual modality during the task compared to the rest. So that means that your continuous performance task enhance the variability in the DMN region. Yeah? So that already suggests maybe there is a relationship between the behavioral variability in the reaction time, which I showed you earlier, and the neuronal variability in the DMN. And that's exactly what I show you here. We now correlate the behavioral variability in the reaction time with the neuronal variability in the DMN. And you see exactly, you see quite nice correlation. And you see the correlations only during the task, not during the rest. So that really suggests that the increase in neuronal variability during the task compared to rest in DMN is related to the variability in your reaction time during the task. So it's not an intrinsic feature of the brain which is carried over from the spontaneous activity to the rest. So to the task. So that really means that the task-related neuronal variability is related to the task-related reaction time of behavior variability. Yeah? So here you can see a direct connection between the neuronal and the behavior level through shared temporal features / variability. Yeah, and we have various control investigations. Now I give you another example. So meaning there may be a temporal shaping, a temporal or dynamic shaping of your attention through variability. Neuronal variability translates into reaction time / behavior variability / attention fluctuation. So here you see a correspondence in the dynamic features of the brain with the dynamic features of the cognition. Here I show you another example of attention. So here we investigated in EEG, so this time it's EEG. We investigated different meditation techniques. So we have a strong collaboration these days with India, and they have really all kinds of very interesting and sometimes a little crazy meditation techniques. It's amazing what they do. And so here we compared 3 meditation techniques. It's the Shunya, Mantra, and Vipassana. Now, these are different induction techniques to get into a meditative state. And these require different kinds of attention focus. And that's what I want to explain to you here. So in the Shunya meditation, you do not focus on any particular object or your body or your breathing. You just let the objects pass by like a river passing by, but you do not touch yourself. So you need and you do not select a particular object. So you need a very wide attention window. The first author of this paper, Bianca Ventura from Italy, illustrated that beautifully. Now, when you do a mantra meditation, where you have continuous thinking, you just focus on the mantra and nothing else. Meaning your attention window is shorter. Illustrated. Just focusing on the mantra and everything else must be basically taken out of your attentional focus. Now in the Vipassana, you focus on different body objects. So you scan, it's called body scanning, you scan this one, you scan this one, you scan this one, this is called mental scanning. So you need to really focus on specific objects, separate from others. I only focus on this body part, but not on my shoulders. I only focus here, but not on any other parts. So you really need to segregate your focus on the particular body parts from others. So now, you already see that we have different temporal widths of your attention windows. Now the question is, do these different durations of your attentional windows are also related to different windows in your neuronal activity, different temporal windows? That is illustrated here. How can you measure temporal windows in your brain? Through what's called the autocorrelation function. Take it for now, I will later illustrate what exactly this autocorrelation function is. Basically, you measure the degree of correlation of the neuronal activity with itself over time. How similar it is. Basically, it provides a temporal window. It's like, and that's basically a time scale and duration of a process. Your heartbeat has a time scale of 1. Your breathing rate has a much longer duration. Here you see the duration of the temporal windows of the brain activity. Now the question is, are these temporal windows here of your attention related to the temporal windows of your brain? This window is long, so you expect a long temporal window in the brain. These windows are very short, discrete body objects, you expect shorter temporal windows in the brain. And that's exactly what we observed. So we observed a very long, delayed, what is called autocorrelation function in the shoe here. Remember, that was with all these objects in the longer attentional window. And we observed the shortest temporal window in the brain in the Vipassana, where you have separate, discrete objects. And the Vipassana has an explicit temporal dimension. You need to focus on this one, this one, this one, and so forth. So because you focus on a sequence in time. But you need to segregate them. Yeah? And that's exactly what we observed here. The mantras intermediate. So here again, you see a convergence of the temporal windows of your attentional demands with the temporal windows of your brain activity. So you will see this back and forth that we saw, meaning the time scale duration, longest here, shortest here, provide a link between the neuronal and the attentional. Yeah? So that's why we speak of a common currency. So, what is the main message? First, I think you can really say neural variability in the DMN, the first study, shapes your fluctuations in attention through corresponding variability. And that's what I want to point out here. Yeah? The neuronal dynamics relates to the behavioral dynamics. And that is what is very important, that usually we average out these dynamic features in our behavioral data, in our neuronal data, when we calculate the mean or the average across time. Yeah? So here, remember the iceberg. The mean is the upper part of the iceberg, the averaging, but below we look for the trial-to-trial variability. Yeah? So there's a temple shaping of your attention by the ongoing temporal dynamics of your brain, or the time windows. See the meditation in the second example. So now I give you another cognitive function, thoughts. So there was one, I got into the thoughts through two students, one of Dr. Long over there, and another student who really got me into the thought dynamics. And now I want to show there's another feature, dynamic feature, is your ongoing phase cycles. Peak, trough, and there's a dynamics to it. And that you link to the changes in your thought. So I show you now the first step, what is phase dynamics? So here I show you a particular way of calculating the phase dynamics. It's called frequency sliding. Peak frequency, it goes back to Cohen and it's 2014 J Neuroscience. And here we applied this actually to the bolt, and you can do this also in EEG, obviously. So, but the calculation is the same for fMRI and EEG. Basically, you take the raw data, you filter the data, then you calculate the phase angle. You do the fast Fourier or a wedge. You calculate the phase angle, and then you take the derivative of the phase angle, and that's basically your frequency sliding. So how much your phase and its associated peak frequency change over time. And here you can see, no surprise, that when you capture the phase in different parts, like peak, trough, rise, and fall, that you have slightly different peak frequency. Yeah. Now the question is, so that's one way of calculating phase dynamics. So the question now, how is that relevant for our cognition of thoughts? You probably know, or maybe some of you even research on it, that phase cycles have been related to the content, particularly in visual perception. The beautiful papers by the group around Gregor Toot in Glasgow. And I remember there was somebody, I think it's at Beijing University or here, where also do revealing that your visual content, your conscious contents are related to the particular alpha phase cycles in visual consciousness. So here we calculated the alpha and theta peak frequency in alpha and theta frequency ranges in the way to track on and off thoughts. What are on and off thoughts? So when you really focus your attention on my talk, and your thoughts are all about here trying to understand on thoughts, then you have on thoughts. However, if you don't care about on and off thought, I said I'd rather think about my meeting tonight or whatever, something else, and I don't care what Northoff says about on and off thoughts, then you have an off thought. Yeah, so that's a simple definition, and you can test it, for instance, with a sustained attention response task, with different tasks for that. You have to listen to certain stimuli, standard deviant, make a response, and then you get also some thought probes. And the thought probes can even say, okay, we have thoughts completely unrelated to the task or thoughts related to the task. And in this paradigm, we also added another thing that we asked the subject, do you have multiple or single thoughts? Do you have many thoughts going on, or is it just one single thought? Yeah, so the thought quantity. So, and then we did, in the same paradigm, we did the EEG-based frequency sliding. Basically, it's a measure of your ongoing phase dynamics. Basically, if you have higher peak frequency, you have a more change in your phase dynamics, meaning your phase dynamics is faster. So it's really a measure of the phase-based neuronal speed. So, and here I show you the main results from that paper. So what we thought, so here we compared off multiple, so we showed, I don't show that here, but that when people had off thoughts, so they were not related to the task, they also had many thoughts going. Yeah, many thoughts wandered through your mind. Typical example of mind wandering. Yeah, you have many thoughts and they just flow there. And then you have on thoughts where you really focus on the thought, and usually that was accompanied also by more single thoughts. So this is by itself already interesting from a cognitive point of view, that your on and off thoughts are related with a certain number of thoughts, multiple or single. So there seems to be a coupling retrieval. So, and then we calculated the peak frequency, the phase dynamics in the alpha range and in the theta range. And you can see here the brown ones are the single alpha sorts, single sorts, and you see a higher alpha peak frequency in the sorts on and single. That makes sense because, as you all know, that alpha is related to the input gating. You look at all the papers by the group around Klimisch, and you can really see, okay, so your alpha is higher doing on and single sorts compared to off multiple sorts with Luga. For theta, interestingly, it was the other way around. So here, the theta of multiple sorts had higher peak frequency than the on single sort. So we also calculated that's the same. You can also do the phase modulus. So basically that you take out the phase angle component, just calculate the pure power, it's called frequency power. And we didn't see these differences in the frequency power. So that's very important because it tells you that these differences between the two types of sorts are really related to the underlying phase dynamics, the phase angle. Because if you take it out, calculate the phase modulus, then you don't see the differences. Yeah, and we have various other controlled investigations. So that really means that your on and off sorts and their contents are related rather to the phase angle and cycle than rather than just the amplitude power. So that really says that unfortunately we couldn't, because of certain methodological issues, we couldn't measure the change in the thought itself, but you said that the phase dynamics, the change in the phase dynamic and the frequency power plays an important role in shaping the contents of your thought as either on or off. And it makes sense. Your theta is slower, so you have a higher likelihood of including multiple thoughts because you have more time. So it's a higher likelihood that you have more thoughts. Alpha is shorter, so it's a higher likelihood that you have only a single thought. And that's exactly what we observe. So this is a summary slide. So you can see here you have an external input, and your thought is the cloud, supposed to be the thought. This is the thinker. I always like this little guy here, and you see that guy is different. So you have an external input, and that is strongly processed. Your alpha comes a little higher, your theta becomes slower, and in your thought, you really focus on the external input, and it's only one single thought because all other thoughts are somewhat suppressed. Now, in the off multiple, you have the opposite modulation. Your theta peak frequency increases, your alpha peak frequency decreases. So you have the reciprocal modulation between alpha and theta frequency sliding, which we also observe in other studies. So that really seems to be an intrinsic mechanism, the way it is wired. And now you see that your thought is all these multiple off thoughts not related to the task. Yeah? So that's what you can see. So now you think about this, basically your ongoing spontaneous thought. That's basically when you don't care about what you're thinking about, all kinds of stuff, but not really much about what I'm talking about. So here you see again, there seems to be temporal shaping of your thought content, on off, through the underlying phase dynamics. So what I want to point out is a temporal shaping, because by considering the phase dynamics, alpha theta, I can distinguish off and multiple, off and on thoughts. Yeah? So there's a shaping of your thought content and your thought quantity, single multiple, through the underlying phase dynamics. So now here is another feature, a study by Dr. Long, who's in the back of the room here. The question, so he raised a very good question, do phase angles mediate the contents of on and off? That's basically an inference assumption which you would make based on the previous data. So now here, how can you measure phase angles? By themselves, you can measure, for instance, the degree to which phase angles synchronize with the timing of the external task, or intertrial phase coherence. So now, when you listen to music, you continuously tap with the rhythm. You don't see me tapping here, but you do this, let's say. You tap with the rhythm to the beat of the music. So you synchronize entrainment. That can be measured by intertrial phase coherence. One of the key persons who developed this was Peter Lakatosh. And here you can see, for instance, here you can test this, for instance, in typical auditory oddball paradigms, repetitive, the same stimuli in fixed intertrial intervals. And you can measure here, you can see a high intertrial phase coherence. So meaning you really synchronize well, your phase cycles, your phase angles synchronize and are very coherent with the timing of the ongoing external stimulus. And here, for instance, you don't see that. That's a schizophrenia, that's a healthy, but I don't want to go into that. So then, Dr. Long did a very nice experiment. He did 2 sensory tasks, 2 motor tasks. I will not go into detail about that. But the first thing, of course, you want to see, is there a difference between on and off thoughts in terms of cognitive function / reaction time. So no surprise, here the blue guys are the off thoughts, and you have longer reaction time in these tasks. The red ones are the on thoughts, and you have faster reaction time. That goes for all 4 tasks, the 2 sensory tasks, the motor task. You see, so this is really a basic pattern. So it's, of course, if you think more about something else than the task, of course, you need longer to be cut. So that's a almost very intuitive thing and replicates the literature. So now the question is, how is this difference related to the phase dynamics, the intertrial phase coherence? And you can see this quite well. So here the tasks were plotted according to off and on, and also easier task, more demanding task. So this is high frequency, which was easier. This is low frequency of the stimuli, and this is more difficult. And what you already can see somehow visually, you really see a difference in the intertrial phase coherence. Yeah? You can see this here even better, that you see the 2 on tasks, the 2 on tasks, the red and the black stuff, they had much higher intertrial phase coherence in theta than in the 2 off tasks. So that's important. Meaning, if I think about the task itself, my intertrial phase coherence is higher and aligns better with the external stimuli. Whereas if I'm in an off-task state, I think about something else, my intertrial phase coherence, alignment to the task, is lower. Yeah, and you can see this here too. And interestingly, I showed you the reaction times before. That also correlates with the reaction times, meaning the longer my reaction time, the lower my intertrial phase coherence. Meaning, if I synchronize better with the external stimuli, I'm more efficient, I can better perform the task, I'm faster. Yeah? I can react faster. If I'm in an off-thought state, I don't synchronize well, and my reaction time is low. Yeah? So here, this is a very nice summary slide. This is probably about the resources. Let's say if you're more in the off state, you think about something else, you have less resources for the external task, for processing the external task. Yeah? If you're more into that, if you have more resources for the external task, then then I have less resources for the internal thought. So it's a balance. And that's exactly what we predicted in this baseline model. So it's not that internal cognition and external cognition are separate, but they share a common baseline, they share resources. And one of the resources is, as shown in this paper, the intertrial phase coherence, the degree to which your external input can synchronize your phase dynamics. Yeah? And that's a balance. So you really have the balance between internal and external oriented is shaped or manifest in its underlying dynamics. And one feature of dynamics is phase coherence. So I think it's a very nice illustration. I think the main message is clear. I hope you can see here phase dynamics shapes your mind wandering on or off task. And we have other papers where we really show that different thoughts have different time scales. It's really fascinating. So, and that's what I want to show you. Phase dynamics is sort of in the background for the specific thoughts and reaction to the task. So, now I'll show you yet another example of the temporal shaping of cognitive functions by memory in collaboration with your group and done by Shen, memory and time scale. So here I introduce you to yet another temporal or dynamic feature. So far we had variability, phase dynamics. Now I show you, oh, I'm not allowed to run out of the camera, I know. Now I show you a third feature, which is time scale. What is a time scale? It's the duration of a process. Your heart is 1 second. Your breathing is longer. Your stomach has also fluctuations, shown by the group around Talon Boudry, around 0.01 Hertz. It's very infraslow range. Yeah? So, and why time scales are important? So this is a paper, which is from a paper in Ticks about time scales. So where we illustrate it. So music has different time scales, slow and fast, an abundance of time. Now, imagine your brain, or let's say your AI device, has only the faster time scales. So it will always dance too fast to the music, because it doesn't lack, it lacks the longer time scales, which are also present in the music. So that guy will always dance too fast. Now, the opposite. You have only the longer time scales, but not the shorter ones. You will always dance too slow. That's the case in depression. That's the case in mania. Real world empirical evidence. So ideally, you have an abundance of time scales here, because that gives you the highest likelihood of matching or converging with the time scales in your environmental context. Yeah? And then you have the high likelihood that you can dance to the rhythm of the music. So that's illustrated. Now, remember I showed you already before the autocorrelation window in the meditation, the 3 forms of meditation. Here you can show how we calculate it. Basically, we have different lags that of course depend upon your sampling rate, the exact nature of the lags, and then you correlate the signal with itself over time. And you can measure that in different ways. At 50%, that's the standard thing. We introduced also the ACW0 where it crosses the 0 line. And there are recent other methods by Go et al. 2022 and 2020 in eLife, where you can also measure basically the shape of the power spectrum after eliminating the oscillatory component. So, what did your group do and what Sven did? So we looked for encoding and retrieval. So now you say, come on, we know that encoding and retrieval are intrinsically temporal. Yeah, but you want to see whether they're really mediated by the temporal dynamics. Of course. And what is important for encoding, you need a long window. Because you enter the room and you want to encode every person in the room. Now I leave the room and you ask me who was sitting at the back of the corner at the left. So when I come into the room, I need encoding. I need long temporal windows because I need to scan you all, and so I need to remember you. Well, that's not good. Now I leave the room and she is asking me who was sitting in the upper right of the end. So now I need to temporally segregate, take you out and just focus on you. So there you see differential, if not opposite, temporal demands on encoding and retrieval. You want to encode efficient, so you need a wide temporal windows to include everything. And you want to retrieve efficient, so I just need to segregate. Yeah, I need to cut you out of this so that I remember you. Yeah, now I certainly will. Yeah, and So that's the thing. So now we investigated these temporal windows in encoding. Yeah. And the first thing what we saw, okay, and we also included the resting state as a control, whether we really want to see is it task specific, because that's important. Yeah. If it's task specific, then you can link it also to the cognitive demands. So here you see the encoding, and you see that the encoding has a longer temporal window as expected than the retrieval. And the two also correlate, no surprise. And here the topo maps, you see also the difference. The more yellow, the longer your ACW, your autocorrelation, and that's exactly what we saw. So then the next step, of course I leave out some of the details here, was that we link the neuronal ACW doing encoding and retrieval to cognitive measures of retrieval. Yeah? So we could, for instance, see if you have a longer ACW in encoding, you have a higher accuracy in the retrieval. Yeah? So if I was really scanning the whole room with one big temporal window, I have a better chance of including the person about whom she asked me to retrieve. Yeah? But if I have a very short window, if I only do half of the room, and then she asked me about a person from the other half of the room, I make a mistake. That's exactly what you can see here. And also interesting, the false alarm rate. The longer my ACW in encoding, the lower my false alarm rate. Yeah? And we have further support. So, and then you see for the retrieval, it's a similar thing. The longer the accuracy, the longer my ACW in retrieval, the better my accuracy. So that really works out well. You can see here basically a summary, but I spare you the details. But what is important, that it seems that the time scale, the duration, your temporal windows doing encoding and retrieval seem to have direct relevance to the cognitive demands of the task. And that's important. Yeah? So you look at basically how they shape it, and that's what these data really support. So, then you say, yeah, then what is the mechanism? We have a temporal window, so what? What do I do when I have a long temporal window and scan the room? I go from you to you to you to you. I integrate you. I connect you. Yeah, and that occurs over time. That's why we can speak of temporal integration. Whereas when I just focus on you, and not on you, sorry, yeah, then I need to segregate you temporally from him and from her. Yeah, I can also change the view. Yeah, so meaning the temporal windows of your brain may be related to temporal integration and temporal segregation. So now, and that is also suggested by all the findings of the Hasson group about the temporal receptive windows, but they didn't show it as such. So how can you show them? So we had a couple of studies, which I don't report here, where in the paradigm itself, there were stimuli presented in a temporal sequence, and the paradigm itself required temporal integration segregation. Again, that's related to the autocorrelation. But even that is indirect. So one way here now is to go to computational model, because that's where we can test this in an explicit way, and that was done here. Let me just give you a little key. So for the experts among you, this is the Wilson-Cohen model. This is the biophysically realistic large-scale models where we enter a DTI matrix and a functional connectivity matrix from healthy subjects. And fortunately, there's a formalism with the 2050 neuron. And we can see, so now, and it models these regions, it models these regions, also their different time scales. So that's why it's interesting. And when you provide a short, so now this is our thing, what we added. So now we provide a short input to V1. So what you see that, for instance, V1, V4 relatively well represents the temporal duration of the input. So the activity, timing follows the timing of the input. now go to a higher order region. So these timescales are relatively short. Now go into 24C, yeah, anterior cingulate cortex. You see the region doesn't care about the different inputs anymore, but it just has one slow increase and then increase. So meaning this is a clear temporal segregation of the inputs. This is the temporal integration. So it's literally, that's the mechanism of, and then we calculated, of course, also the autocorrelation windows for V1 and 24C, and you could see 24C has a very long ACW, no surprise. You integrate the different inputs. V1 had a very short ACW, temporally segregated. Now you say maybe this is just related to your input structure, so now we also did the same for the long input. Same thing. Your V1 represented the duration of the input very well. 24C, very slow increase, meaning it's on a much longer time scale. There's a lot of temporal integration going on here, temporal segregation. So you can see we have other papers where we combine that also with other psychological studies. So here you can really see that maybe during the encoding, we have strong temporal integration of different inputs. Whereas in the retrieval, you need to have temporal segregation because you need to focus on one specific item that you were asked to retrieve. Yeah. So, then the next question is, so now I show you timescales can also be measured in the frequency domain. So then the question is, what are the mechanisms through which the input, what features of the input get into your memory? So this is now a different study. This is an fMRI study. And now I show you how the path from input to memory. In the first, it's implicitly in the encoding, now we do this explicitly. So this is a thing what we did here. So we calculated this in an fMRI study. And you could see, we calculated the behaviorally the capacity of the subject. So basically, we had this staircase procedure and determined the capacity, how much it was a visual memory task, as you can see here, with these dots. And then if they made a mistake, the load was decreased. If they will succeed, it increased. So we really look for the cognitive capacity of the subjects. So, and what you can also see here, one trial duration was 9.5 seconds. So why is that important? So when you work with me and you have a task paradigm, the first thing you will see, I will ask for the temporal structure of the paradigm. What is the stimulus duration? What is the ITI? And so forth. Yeah, so here we had one trial, 9.5, so there is a certain task periodicity. That's important, and you can recalculate that in terms of frequency, meaning in 0.1 Hertz. And then we also had a control task where we did not have any such task periodicity. So why do I say that? Because maybe the task periodicity is important for the encoding of the input into memory. So this is important for the methodological side. So we could capture the 0.1 Hertz because we could set a bandpass in the fMRI from 0.1 to 0.45 Hertz because we had a TR of 1.1. So this is important. If you have the usual TR of 2, it will not really not capture it. So now we said, okay, how can we measure that? So is the temporal structure of your paradigm reflected in the temporal structure of your power spectrum in visual cortex? So that's basically what you used from EEG in the 40 Hertz domain, the steady state potential. Basically the same principle when you have a 40 Hertz ASSR or BSSR paradigm, you basically get a peak in the gamma range. So same here, you get a peak now in the 0.1 Hertz because that corresponds to the task paradigm frequency structure. And you can clearly see this in the one, very nice, you can clearly see the peak at 0.1 Hertz. And interestingly, you see this peak very well in the visual cortex. but you don't see it anymore in the dorsolateral prefrontal cortex or the hippocampus. So as you can hear, this is illustrated, and interestingly here, your peak, your brain, this you can really see. We also showed the same, and that this was not present in rest. So you could really see also here in the control. In the control, you don't see the peak. Here the control is all the same. Here the peak is really different. The blue stuff that really tells you is related to the temporal structure of the task. And you can see this here again. And we calculated the median frequency. The median frequency is basically the average frequency of your power spectrum. And if you have a low MF, median frequency, it's shifted more towards a slower frequency. Now the question, how is that encoding of the temporal structure of the paradigm of the input stochastic related to the cognitive performance? So here we could see, now we correlated the peak in the V1. Remember the peak there, it's the 0.1 hertz, the task frequency, with the cognitive capacity, the working memory capacity, and you clearly show a very nice correlation, which only holds in visual cortex, not in the other regions. And you can also see here we distinguish between high and low performers, and you can high peaks and low peaks, and you can see again the relationship with different capacities. So the main, and then interestingly, in the hippocampus, and you want of course hippocampus memory, I don't need to tell you that. So here we observe it's not just a local peak like in V1, but here the shape of the whole power spectrum. That's why we speak of a global power spectrum relates to working memory capacity. Yeah, so and we showed this in various control investigations. So here it seems really the local in V1, the local, how much you encode the task periodicity at the local target frequency. Here it's the global shape of the power spectrum during the task, which is relevant for the cognitive performance. So here again, you see a temporal shaping of your input and your memory performance, yeah, through the alignment in your power spectrum. So meaning the temporal structure of your input is very important for the brain because it picks up that temporal structure, and the better it picks up that temporal structure, the better its cognitive performance. So you see, you basically trace how important the input encoding is, or the input processing. That's, for instance, in the encoding and retrieval paper, Shane also very nicely analyzed the input itself, the complexity. So we are now analyzing more and more the input themselves. So, main message, timescale shapes the processing of inputs to memory through possibly their duration and speed. So the duration and speed of the inputs. So, and again, so you seem to see a certain corresponding that your timescales shape your cognitive memory. So it's almost trivial because memory is intrinsically temporal. So it's the temporal features of your input and the degree to which your brain encodes these temporal features of the input shape your memory. Yeah. So, last thing, a very hard nut to crack, I didn't go into too much detail, is consciousness. I so far only showed you temporal features. I did not show you spatial features. Now I show you two examples of topography and spatial features, how they shape mental features. That's why we speak not only of temporal neuroscience, but spatial features. So, I will not go into consciousness and detail. So first, what is topography? When you look up, topography comes from geography. It's a form and feature of mental features. It's lines, what you see on maps. Same thing. So what is important here is the relative relations among different brain regions. For instance, I'm a big fan of this unimodal, transmodal gradients, the Magula stuff. And actually you can show this also for timescales. We have also a timescale gradient. Unimodal regions have shorter time scales, transmodal regions have longer. This is from our review paper, but also from other papers, and has been shown by other people too. So now here I show you how is that topography, unimodal, transmodal timescale differentiation, relevant for consciousness. So here I say maybe your consciousness is shaped by such a topographic distribution. So here we show again, this is a paper from 2023, in different states. So this is awake, sedated, anesthesia. Same subject. And we here calculated the power law exponent, is basically the shape of your power spectrum, for the core regions, transmodal, and the peripheral regions, unimodal. No surprise, the power law exponent is slightly higher for the periphery, and that holds for both awake and sedated. It no longer holds in the anesthetic state. So you have the topographic de-differentiation in the anesthetic state. And I will show you more. Now, then we did also a task paradigm, and here we did a task paradigm in these states with blocks 25 to 35 seconds. So again, we had this task periodicity. So in this, you can see here, the blue guy, that's the task, that's the frequency of your task. And you see when you look in detail, you see some power spectra peak in exactly that frequency. Now, in the anesthetic, even in the sedated state, in the anesthetic state, that peak is no longer there. Meaning, when you're anesthetic, you don't care anymore about the task structure. You do not process the input anymore, / the temporal structure of the input. And what else you see, this is very interesting, that your power spectrum is completely flat. It's no longer scale free. You don't no longer have this distinction of slower frequency having more power, faster frequency having less power. Yeah? It's completely flat, meaning, and that's important for the next slide, this is white noise, this is pink noise. Yeah, very important. Your environment is full of pink noise, not white noise. So, I show you here the topographic differentiation when you lose consciousness. Here are the different networks according to the power law exponent. You can see this. This is rest, this is task. Here you see the hierarchy. And even in the sedated state, the differentiation is still there and you still see the hierarchy. Anesthetic, it's all the same. So you really have a topographic de-differentiation. Meaning your different regions and networks in the brain are very important because they all contribute something slightly different. Yeah? But your brain becomes the same. So my imagination is that in this state, your brain is basically completely no difference anymore. It's one homogeneous soup. And that's why you don't have consciousness. You don't experience anything in your consciousness anymore. It's over. It's just one dark black hole. Yeah? So, now, why is the brain spin noise? So here, we really focus, you see this is some of the studies with several others, they really focus on the input encoding. So why is that so? So here we use the computational model, the Stuart-Landau model, the simplified, these two oscillators. You can see oscillator here, oscillator 2. And what we also did, we provided different kinds of input. So we provided white noise input, pink noise input, brown noise input. And we measured the effect of this on these two oscillators through phase space attractor dynamics. And you can see the more you have movement here, the more the system reacts to the input. If you have a very homogeneous like here, there's not much reaction to the input. So the indirect measures. You can see that also here. The perturbation, you need a lot of, here, even if you increase your noise strength, your white noise will not change much in your oscillators, in your model. So meaning, and you can see this here again, if I have higher noise strength, your pink range is optimal to react, and on the other hand, to keep stable. Whereas white noise, you need a very high dosage of white noise for your system, to your oscillators to react. Meaning, when you are confronted with white noise, your brain will not react much. Yeah, and that's, you know that. There's this white noise things when you can't sleep, just don't make some noise. You can sleep, but you have white noise. So your brain is tuned to pink noise. And if you lose that tuning to pink noise, you lose consciousness. Yeah, and this is, I always like this figure, first author Philip Clark did this. Here is the pink noise. Yeah, the pink noise in your brain makes it able for you to react to the pink noise in the environment, like the temporal structure of your task. But if you have white noise in your brain, completely flat power spectrum, you cannot react. So meaning your pink noise, your scale-free dynamics is very important for the input encoding, and that's very important for consciousness. So that's one example of consciousness and topography. For the matter of time, I probably skip here the depression because I don't want to put you in an off-thought mode. So I skip that, but I give you another example. For consciousness and topography, where you can see this label is meditation. So this is about an fMRI meta-analysis of proficient meditators. And one key feature, I'm sure you all know this, because you're in the Asian tradition, is non-duality. Meaning you don't make a difference in your experience anymore between the outside world and yourself. You experience yourself as part of the wider world, and you don't make a difference anymore between yourself and the other. It's like the psychedelic effects we had this morning in Ketan. Yeah? Typical and proficient meditators discussed a lot. Yuzipovich has beautiful papers on non-duality. And I think here the first author of this paper, Austin Cooper, illustrated it beautifully. So here you have a real distinction between environment itself, and here it's just non-duality. And that's a typical feature. Many subjects report that, the non-duality. Now, so what happens in the brain? So usually, and here this is exemplified, and this is data-driven, this is meta-analysis, that you really have, you probably all know this, the well-known anti-correlation between the default mode network, the blue stuff, and the central executive network. So in a way, and you also have the topography between transmodal and unimodal. So that's sort of a dual topography, yeah? Particularly the anti-correlations. And as you all know, particularly the midline regions DMN have been associated with self-awareness, self-consciousness. So that's basically put in contrast in your topography to sensory, the environmental. So you have sort of a dual topography. But what you see in the meditation people, in the proficient meditation people, suddenly you don't have this anti-correlation anymore. You see this here. You don't have a negative correlation, you don't have a positive correlation, you have a complete balance. Yeah? So, and your gradient of your unimodal and transmodal topography also flat. And the same happens in psychedelics. We just submitted a paper exactly showing that. So meaning your dual topography becomes non-dual topography. So now we assume that that non-dual topography is also related with your non-dual experience, where you don't make a difference anymore between self and environment, between internal cognition and external cognition. Yeah? Because the topographic structure of your brain doesn't allow for that differentiation. Yeah. So then we did the same for these meta-analyses for the three-layer self-regions. So what we saw in the novel meditators, there's a lot of activity in the mental regions of the self, but low activity in the inter-exoceptive. Now, in the proficient meditators, you had not much activity here, but strong activity in the inter-exoceptive process. Meaning you're better aligned and connected with your environmental context. So now the next step, of course, we're trying to have measures of non-duality and links to the duality of the topography. But I can say this is strongly confirmed by subjective reports from psychedelic studies. Yeah, exactly the same thing, similar change in your topographic structure, change in the structure of your consciousness internally. So that's why we like to speak of a dual versus a non-dual topography in dynamics. Yeah, and you also, you might not have noticed, I always emphasize here this temporal spatial alignment, how the iceberg aligns to the water. That's basically how your brain encodes the environment. That's really key, and of course also the body. Yeah? So that's why we speak here beyond, I think I like, he came up with the title Beyond the Way of Reality. So, and you have a topographic reorganization. That's something we observe also in depression in a different way. There's a topographic reorganization on the neuronal level. And that goes along with a change in your mental topography too. So because you experience self-environment in a different relationship. So again, let me say topography changes, shapes the structure of your consciousness. So, conclusions. I hope I could show you some of the relevance, but you might say, why is all that relevant? You all knew your temporal and spatial stuff is trivial. That's what I heard in many reviews in the early days. And dynamics and topography are trivial too. So how do you make the leap to the moment? We are not complex. So, and I think in that moment you might want to take a step back and look into the models of your cognition and your brain. So, as I understand, please correct me if I'm wrong because you're the expert, the predominant background model of your cognition is basically you have a stimulus content, you have a sort of a foreground, then you link for a neural correlate, event-related measures, for instance, and then you have a behavioral foreground. So, but one thing which I think is often taken out of the data is the intra and intersubject variability. So, where is that coming from? And that's one thing when you analyze data with me, it's the first thing I will ask for intra and intersubject variability. And I'm not asking that to say your data are not valid. I'm asking that for the opposite reason. If there is no intra-intersubject variability, I will not believe you the data. Yeah, because, and you all, everybody who deals with data knows that. So I take that very seriously. So how do you account for that? So one of the reasons where we say, of course I cannot go into detail now, is that we suggest maybe it's better a background foreground model. Your background is your continuous, spontaneous, dynamic, and your topography. And your foreground activity are all your standard measures. But you have a strong impact on your background activity on the foreground. For instance, we have a paper coming out where we really show that ACW predicts your event-related potential. Yeah? So you have a strong influence. And the inter-individual variability is coming from people. It's coming from your spontaneous activity. So that's why we have a strong line of research where we really look for the interaction of spontaneous activity and task avoidance. Because that is our pre-stimulus and task. We have a paper in Physics in Life Review very recently where we really show the pre-stimulus activity shapes the contents of your consciousness. Yeah? And that's important, this is not just a linear superposition between spontaneous activity and task avoidance. It's a non-additive, non-linear mechanism. So this is not trivial. And that's key for your consciousness. If you lose the non-linearity in that interaction, you lose consciousness. So, now, imagine, I always like this analogy, so paintings. Leonardo da Vinci is a master of background and foreground. Here, this is very important. If you don't have that background, Mona Lisa looks completely different. So he's a master in linking background and foreground. Your Chinese traditions, you're even much better master than earlier. Your background, you often have these mountains in the background, and then the river flowing and the people in some foreground. So you have this background, foreground. Now, the two belong together. If you isolate the foreground from the background, you don't see that anymore, and vice versa. Same here. This is the Last Supper by Da Vinci. This is the original picture. Now you take out the foreground, now you take out the background. Doesn't look anymore. Yeah? So that's exactly key. And you also see here, the background is very important to place these 12 apostles in a three-dimensional context. That's exactly what the background does for your consciousness. You process a single input, and through its topography and dynamics, your background probably puts that single content input into a 3D virtual space. That's your mental experience. That's your consciousness. So, putting it all together, I showed you how different dynamic features impacted different features of cognition. Temporal spatial alignment, that was the consciousness stuff. Intrinsic neural timescales was the meditation. Neural variability, the attention. Phase dynamics. And you already see what I now did here. This is important. This goes from longer to shorter times. That's why we develop a dynamic layer model. Dynamic layer model, basically, I conceive the brain radically in terms of timescales. And how these timescales, now next picture, now you can have a psychological view of the same model. Now you have content, remember the thought content, or perceptual content, attention, memory, consciousness. Different timescales. So I consider cognitive and mental features radically in terms of time and also in space scale. So that's why I would argue that the dynamic layer model of brain is an integrated brain cognition. So, why is that important? Second to last slide. I already indicated that because that's what is shared between neuronal and mental activity. This is a very recent review paper where we summarized many findings, where we can really show, okay, the speed, duration, variability links neuronal and mental. Same in the topography. And that's why we speak of a common currency. So now, that's the second to last slide we will really stop. You say, yeah, Northoff, but it's not as easy. You don't have a one-to-one relationship. It's not that the time scale of your neuronal process exactly matches the time scale of your cognitive process. Right, indeed. But you have a complex correspondence. For instance, when you do transfer entropy from neuronal to behavioral time series, you can see a certain lagged version. So there is a causal relationship, but in a lagged, temporally lagged way. That's why we speak of a continuum of simple versus complex. And we use some of that also for mental disorders to diagnose in therapy. So, why shall I care about spatiotemporal neuroscience? Is it yet another brain-mind theory in for failure? I hope so, no, and you can prove me wrong. So first, I think it can really better account for intra-individual variability in the data. We try to get, and I discussed it with some of you, the neuromental mechanism, specifically neuromental or neurocognitive, not just neuronal mental. Yeah, how neuronal activity transforms into cognitive or mental activity. And as I said, I didn't show this, we develop based on that biomarkers for diagnosis and therapy in mental disorders, depression disorders, speech disorders, and so forth. And with that, I'm closing, and I hope that I could convey the message of dynamics and topography connect the brain to cognition. Thank you.